pleased to introduce Dr. Hassan Abolhassani, which is working in the Karolinska Institute in Stockholm, and he will present approach to the genetic analysis of non-monogenic antibody deficiency.
Thanks for the invitation, and I'm really glad to participate in this G Project Congress again after 3 years in this fantastic country and beautiful city, Konya, the city of the philosophy and the spiritual intelligence. And of course, I want to also appreciate all of the kindness from the organizing committee and scientific committee. As the Maulana said himself, from the kindness, the spike will turn to the flowers. So thanks everyone for this fantastic atmosphere. For the people who are following our series of the reports from the antibody deficient patients, I'm going to do some updates from our recent works on the cohort of the antibody deficient patients. Today I will focus only on our cohort from Iranian patients, but maybe I refer a little bit also about our Swedish cohort as well. So, I think I'll disclose. Of course, we know that antibody deficiency, among all other 9 different categories of the primary immune deficiency, are the most common one and actually has the highest burden of the disease. We consider them as a the second most common immunodeficiency in the world after the HIV infection, so we should get more aware about them. And that's the reason I want to start with the classification a little bit. Maybe a majority of you know about this, but let's have a kind of similar words at the beginning. So we have 4 different categories of the primary antibody deficiency. We could have a defect in the early B-cell defect, which usually present with the agammaglobulinemia, which means the reduction of the old immunoglobulin level in the periphery. We could have a kind of terminal B-cell differentiation problem, which we define them later on as a common variable immunodeficiency. So basically the plasma cells cannot be produced, and then the antibody level, all different class of the antibody, mainly IgG and IgA and IgM mainly, could be reduced in the periphery, but the B cell in the periphery are normal. We could have a class switching recombination defect, which usually present with a high level of the IgM in the serum, and we call them CSR defects. They also have a reduced level of the class switch immunoglobulin IgG, IgA, and mainly IgE as well. And later on, the last group of the antibody deficiency are the light chain defect, or the major disorder in this category that we may all know are IgA deficiency. So basically each defect in the development steps of the B cells could lead to agammaglobulinemia, CVID, hyper-IgM, or subclass deficiency, or selective IgA deficiency. But the part that I realize in the field that the majority of the people are a little bit unaware about the history, and actually the history is one of my favorite topics, so let's start a little bit about how we come through this stage about the antibody deficiency. So we know that the first case of the immune-related disorder has been described many years ago, almost 1,000 years ago, by the Hakim Razi. First case of the asthma and allergy, and later on with his second generation student, Abolhassani. He also described the acquired immunity theory. In that sense, is also associated with the humoral immunity that we described it later on. But there was a gap actually in the Middle Age. There was no study further on about this field of immunology till 17. When with the efforts of the Edward Jenner and Louis Pasteur and Rox, we had a kind of feeling about inoculation and vaccination that our immunity can be produced and it can be acquired to the one person and can be secure, an immune system being defined at that time. It takes actually 115 years later on that we come through the stage that the first case of the antibody deficiency has been described by Broughton in 1952. But it's also nice to mention that the first Nobel Prize in medicine also received by the Emil von Behring on the he actually defined the theory of the antibody formation and the transporting to the other person by including some interesting instrument that time, centrifugation. He produced a pure serum, and then he shows that if you transfer those part of the blood, you can make a secure person for the diphtheria or tetanus. Then we had a pure antibody preparations by the help of the electrophoresis, when the electricity become available in the field. And later on, we had the antigen-antibody binding hypothesis in 1934. It's also nice to mention that the first case of the immunodeficiency has been described in 1926. In the second one, we had a pure antibody purification by the French group, but that time we just know about the neurological manifestation of, because ataxia also has some humoral immune defect. But they didn't recognize the immune system problem. We can say the pure association of the immune system defect with the infectious disorder has been described by Hassan in 1950 for the phagocytosis disorder, and then by Broughton, the first case that we know about the antibody deficiency. Just to go fast a little bit on the history to today, that we know where we are standing. So after the first case of the XLA being described in 1952, very fast the other cases of the antibody deficiency has been described by the Janeway in 1953. We know the CVID, IgA deficiency by the German group in 1957. We had hyper IgM syndrome by the Rosen, and also hypo IgM syndrome by the John Raymond Hobbs. We had also the IgG subclass deficiency defined in 1970, and later on a little bit molecular studies being better by the animal models. We know about the antibody structure, VDG rearrangement, and the origin of the B cells, of course, was not defined in 1974, that the B cells coming through the liver and bone marrow. Then we performed the first gene discovery finally, which is nice to know that one of the first genes that has been described in the field of PMID was actually associated with antibody deficiency. We know the kappa change in 1985 in the science paper that you can see here by the Ben Zerger's group. And of course, then molecular study being more available and the people knows about the BCR pathway, the B cell receptor signaling pathway, they try to, by targeted level, find the gene location. And before the 2000s, before the beginning of the century, we had mainly the discovery of the many genes associated with the BCR signaling pathway. So BTK, IGHM, IGLL1, CD879A, and BLNK. And we had a few also feeling about the hyper IgM syndrome, the class 3 gene recombination in 1993. We know about the CD40 and we know in the 2000, by the releasing of the database of the human genome, we had more advanced technologies and genomic tool for approaching to the field of antibody deficiency for the cases who still have being undiagnosed till that time. So more genes has been discovered by class 3 gene recombination defect, AID, CD40, and UNG, and later on PMS2. And in a specific field of the CVID, the terminal B cell defect, we had also discovered ICOS, HACHI, and BAFR, and the CD19 complex disorder. And we know after the 2010, by coming the next generation sequencing available, using the next generation targeted sequencing or whole exome sequencing, whole genome sequencing, we have a very fantastic tool to basically approach all undiagnosed patients, which is useful, but the topic of today is that how we can handle the cases that cannot really solved by these today available methods. sorry, I want to just mention up to now we have 14 genes defined by the iOS 2019. The report will become soon in the Journal of Clinical Immunology, but just you can see some new genes that are not really being before in the iOS 2017. And if we want to just put them in a diagram of the B cells, crosstalk with the T cells, let's consider this a T follicular helper T cells. I mean, this is a little bit unupdated picture, but we can say 90% of the genes are located in the correct position. And the thing I want you also consider that in 90% of the patients with the early B cell defect, basically BCR pathway, and 90% of the cases with the CD40 ligand, CD40 plus gene combination defect, they have actually the correct genetic diagnosis and molecular diagnosis, but our main problem are with the cases with the terminal B cell defect, the CVID, or later on IgA deficiency and subclass deficiencies, because you can see how spread are the molecular defects underlying this disorder, basically any molecules in the T cells or B cells that are important for the germinal center functions or the plasma cell formation, and apoptosis are actually important and could lead to the phenotype of the CVID. So it's very broad, very difficult to diagnose, and that's the reason the next generation sequencing the recent years help us for diagnosis. So let's go back to the epidemiology. We know that the primary antibody deficiency, as I mentioned in my first slide, are not only in our region, but in the globe also. Near to 50% of the cases that has been discovered by many different centers, by Jeffrey Model Foundation centers, are belongs to the group of the antibody deficiency. This picture also shows the schematic distribution of the disorder. Of course, the countries with the higher level of the consanguinity marriage, mainly in the Middle East, they have a little bit reduced percentage of the antibody deficiency from those countries that have national registries. But in the Western countries, we have some countries like Australia, some Nordic countries, over than 70% of the cases with the antibody deficiencies. Now I want to narrow down to our Iranian cohort, as I mentioned, because the most result that we present are belong to this cohort. We have now, this is the report that we published in 2018. The expected number of the patients with antibody deficiency, if you consider IgA deficiency also, should be one case from 650 cases, which means we should have in our country near to more than 1,000 cases, but we only discovered 900 cases, which means, I mean, of course, majority of cases are unsymptomatic. That's the reason we have just hospital-based approach to discovering of this patient, but from those 900 cases, we successfully sequenced near to 200, 300 cases, and from those we solved near to 100 cases, which means 12% of the patient with antibody deficiency are now solved from the whole 900 cases. Yeah, the registry is actually referral centers, supporting by 31 centers around the country. The people who are involved, the subspecialists working with us, supporting us with the registering the patient in the online version. Now we have more than 4,000 cases in different all 9 categories of the primary immunodeficiency, and we're also mirroring the registry of Iran in the ECID registry. This is the approach that we are considering for each patient that coming with the primary antibody deficiency signs or symptoms. Could be atrophic tonsils, could be the special manifestation, the upper respiratory or lower respiratory manifestations, but we also aware about the other non-infectious clinical presentation of the patients like CVID, lymphoproliferation, autoimmunity. We also consider all of those manifestations. If we suspect some patient with the primary antibody deficiency, there are 2 options: the patient is dead or alive. If the patient are alive, we consider first if the classical clinical presentation are agammaglobulinemia or hyper-IgM, we just directly go for the targeted sequencing. As I mentioned, the number of the genes in the agammaglobulinemia or hyper-IgM are very restricted, so it's very easy to diagnose them with the targeted Sanger sequencing, for example. But for the cases with the presentation of the CVID, IgA deficiency, hypo-IgM or subclass deficiency, we go directly to the next generation sequencing, whole exome sequencing. And also the cases who are still unsolved after the Sanger sequencing with agammaglobulinemia or hyper-IgM will also include to our study on the whole exome sequencing. There are some references for you if you want to know more about the detail of the methods that we work. Just summarize the pre-step for each patient's sequencing, which should be considered, and do not blindly sequence each patient. We should have a complete demographic, clinical phenotype, immunologic phenotype of the patients. Better that we have also radiosensitivity in our opinion, also T-cell subset, which help you in which step of the B-cell development your patient could have the defect. This is also the complete set of the assays that we do for our patient, but I want to just mention the beginning approach or the start point for each patient is the genomic approach. So you should start the Sanger whole exome sequence. This define where you should go further for the transcriptomics, proteomics, microbiome, or even the immune repertoire, for example, assays. You should know the genetic background of each patient, and you should perform it unbiasedly for all different categories of antibody deficiency. Maybe some of you missed this paper that we just published this beginning of this year. Actually, it's available into 1.5 years. I just want to highlight some important points that we think the approach to antibody deficiency should be performed based on this clinical guideline. So please consider all manifestation of the antibody deficiency, which means autoimmunity, enteropathy, lymphoproliferative, and malignancy should be considered. Please do check all of the pedigree or relatives of the available patients, at least first degree, because we know that the CVID, IgA deficiency, subclass deficiency can be clustered in just one specific family. And if you have more patients that are very useful for finding the correct diagnosis, please perform all B-cell and T-cell subset, which is available in your center, which I will tell you why it's important to perform this kind of pre-studies. And please check all patients every 6 or 12 months, because we know that the patients could be progressed to the other form of the antibody deficiency as well. After this data, we are performing, as I mentioned, based on that method, we should consider also sometimes the chromosome abnormality and cytogenic defects, because we know that the four chromosome monosomy or trisomy 8, 10p, some ring chromosome, like ring chromosome 21, this cytogenic defect also can be underlying of the antibody deficiency. So do not forget cytogenic assay. Later on, if you perform all of this step, you can go for the next generation sequencing in case of the unsolved cases, unsolved agammaglobulinemia, unsolved hyper-IgM, and common variable deficiency, IgA deficiency, and so on. And we do a special pipeline that we have for our analysis, but still we know after this all steps we end up with some idiopathic antibody deficient cases. This slide is so small, you cannot read, of course, from the far. Just want to mention the number of the genes that are highlighted now in IOS are just the full preference of the genes that intrinsically affect the B cells. But the patient that you see in the clinic could be affected by 200 different genes. We just summarize our data in the red, and the other groups, coming from around this Helen Chappell groups all together. This number of the genes that basically all 9 categories of the antibody deficiency, primary immunodeficiency genes that we define as a full preference of the, for example, combined deficiency, syndromic features of the autoimmune deficiency, can all have some gene defect that present only with CVID, IgA deficiency, or antibody deficiency. That could be the only immunological manifestation that you can see in your patients. So please do unbiased next generation sequencing. That would be helpful to not only narrow down the antibody deficiency genes associated list, you can also consider the other 200 genes as well. So as I mentioned, the immunophenotypic is very important. We could not find any genotypic and phenotypic association in our families. We have many different discrepancies from the clinical phenotypes, even immunoglobulin level 4 or 5. The only robust immunological phenotype that we confirm in the majority of our patients is that actually the B cell pattern, which will tell you where is the arrest of the B cell development, could be helpful for the genetic diagnosis. So basically, the patient will have a DNA repair defect. You can see the VDGR regimen and class 2 gene combination defect, but still you can find B1, for example, the pre-free. The patient will have some kind of co-stimulatory problem, like a BAFF-R touching, will have a defect or reduced level of the B cells in mainly all kind of the peripheral B cells. And so on, you can find a T cell stimulatory defect like a CD27, CD70 deficiency at the level of the T follicular helper T cells. Support in the cases with the LRBA or CTLA4, you find just in the last part that are producing the plasma cells. So immunophenotyping actually could be very helpful and useful tools before going to genetic diagnosis or for kind of confirmations in the level of the immunologic studies. Why we should do that? Because it's very important. Actually, 50% of our cases could have benefits from these studies. Some cases with the misclassified as antibody deficiency, still they have some underlying combining deficiency, needed transplantation immediately, as we find many cases with the RAG1, JAK3. Maybe you saw some of those reports before associated with CVID. There are some cases with DNA repair that they should avoid having too much radiation or malignancy triggers. We have some kind of targeted treatment for the patients now for LRB, PI3 kinase pathway defect. And of course, we could help patients. The majority of these families still are in childbearing age, and we should help them with the correct familial consultation. Yes, we also conclude with our monogenetic part that the unbiased approach is necessary for all PID patients, not only for CVID or CVID patients with complicated cases. No, you can define very easily for majority of cases with only infectious presentation. Also, the genetic diagnosis should be unbiased, so whole exome sequencing is less complicated for analysis, and also kind of cost effective for a moment. Of course, in future, maybe whole genome sequencing will be replaced, but for a moment that we are talking whole exome sequence, the best options. We know that with this approach, we can find some new phenotype. We could find some new inheritance pattern, or you can find also some novel genes underlying the antibody deficiency, which is very encouraging. And this is not only also, we know that the genetic background, for example, in this case, Iranian, could be solved easier or better because of the consanguineous marriage. But our study, I just want to refer now in our Swedish cohort, we could also solve with this method 40% of the cases with antibodies, any kind, IgA deficiency, hyper-IgM, CVID as well, in the Western cohort as well. So now we're coming to the stage that we are know about the monogenic disorder associated with primary antibody deficiency, but what we should do with those idiopathic cases. So many people suggest you should perform whole genome sequencing. Maybe we heard some of our friends before, and this morning we had also some comments that we should go for whole genome sequencing. But we did this study before. We approached some cases from the beginning, whole genome sequencing, in parallel time that we do some whole exome sequencing for other cases. But we see a very, very little marginal zone effects between the solution part. So basically, if you go further for whole genome sequencing, maybe it's not a very good option. But still, the approach is open and many people can do that. Still, we also, for some unsolved cases, we perform that, but there are some other cards on the table that we should consider them, some other solutions that actually this theory comes earlier to the study. Just want to mention two approach, which is the GWAS study or polygenic score and HLA typing. The few minutes that is left from my presentation. So HLA, background of the antibody deficiency has been discovered for many years ago, even before the time that we have a very well-defined BCR pathway or B-cell intrinsic signaling pathway definition, because many people know that the MHC is important for antigen presentation and for the recognition of the T follicular helper T cells, not only MHC class 2, but also MHC class 1 for the confrontation with the natural killer cells for the presentation of the polysaccharide antigen as well. And we know that many patients with a further clinical study that CVID or IgA deficiency could be associated with many autoimmune disorder, which we know that the majority of these autoimmune disorders are also associated with the MHC regions. So that was the beginning thought of the many scientists in the field in 84, Swedish group, Lennart Hammarström, my supervisor in the Karolinska Institute, he started many years ago to have this hypothesis HLA antigen could be the underlying susceptibility for IgA deficiency. Later on, American group, Dr. Schaffer also had this hypothesis in 1989 with MHC class 3 associated with common variable immunodeficiency. In the 10 years before that time that we come to the release of the biobank or database of the whole human genome database, many study or nice study, at least 10 of them I gather here. You can see perform different kind of assays on HLA-DQ, in the HLA-DQB, and majority of the study at the 2000 country did this a special haplotype that we call it haplotype 81, which is a B8, DR3, DQ2, which is associated with both CVID and IgA deficiency in families that have a kind of familial clustering of the patients. That kind of hypothesis was silent for a period of time, if you can say that, at least for the 10 years, till this paper coming out also from our group, the Hammarström group, that was published in the Nature Genetics, 430 IgA deficiency, which was compared with the 1,090 cases from the same population, Swedish population, and we performed the GWAS study. You can see nicely the first hint in the GWAS study on IgA deficiency in this level. Of course, we had another non-locus associated with the chromosome 6, IFH1 in that case, but you can see the first hint in IgA deficiency was HLA-DRB1. There was 2 other studies, mainly in the common variable deficiency group in the Philadelphia working on that, Dr. Hankerson, and they also showed the 2 consequences of the study, one with the 363 CVID patients, MHC is the first hint, and another study that happened in 2015, same group, they showed that the ITAGM1, because of the bias that they have before, they tried to decrease the number of the CVID patients, they reduced a little bit the number of patients, which I address in my next slide. 164 pure CVID patients, still MHC class region, are in the second top hint after the ITAGM. So why we think those studies are important but still biased? Because they didn't reduce the effect of the monogenetic disorder. So when they called the CVID patient, there was not important in those study, in those which was before 2000 studies, early studies, and also those majority of those GWAS study, they didn't consider them to reduce or exclude the monogenic disorder, which we think is crucial. We perform with our cohort, Iranian cohort. We use the same data that we had for the whole exome sequencing to perform HLA-4 resolutions. The method module is the optimum, OptiType, which has the accuracy of the 97%, which means if you perform also in parallel full resolution Sanger sequencing, which perform also in our selected patients from our cohort, you will see 97% of the data which is generated from the whole exome sequence is correct. If you have coding regions, you can go to the 3 digit. If you have some good coverage also for the non-coding region, you can go still for the 4 digits. And with that method, we compared our monogenic disorder with the non-monogenic patients, or we can say unsolved cases. We have 2 approaches for that to see if there is any haplotype associated with the disease or are more enriched in the patient with which they are not monogenic. Actually, we had a fantastic one, which is a W1, DMA1, DMB1, and TAP1. This haplotype, which is very enriched in the cases who are not defined with the monogenic disorder. But there is a second approach, which is also important to be considered. We think that maybe a specific HLA is important for the antigen presentation, so any kind of the combination of the HLA should be also considered. So we do a regression model to see which specific allele could be predict better the non-monogenic disorder, not only haplotype, that all allele will be together. So any kind of combination of the HLA could be associated. You can see B35, DQ1, and DQB13 are actually enriched in the patients who are non-monogenic, and the 3 other are enriched in the patients who are monogenic. So if you consider this combination of the 6 HLA, they can predict very well for you the patient could be monogenic or not monogenic. You can say it's 6 allele if you just perform a specific sequencing for those regions, we can easily detect the non-monogenic form. But to consider more what we can do further in the genetic approach of these patients, we continue to perform a kind of GWAS study, but in the exome level. So we perform, usually when you do a genome GWAS study, you do a linkage disequilibrium, which means that you want to find specific regions that could carry the underlying genetic defects. But there are some other methods that we call it polygenic score, which we consider all SNP as a player role in the model. So it's also a regression model, so you can consider all of the SNP that could predict better the non-monogenic versus the monogenic ones. And then you can perform with this exome data and the SNP who are really enriched or significant, you can perform a Mendelian randomization. What is Mendelian randomization? To make a long story short, we know that the SNP could be associated with the, for example, in the case of antibodies with the immunoglobulin level, and SNP can also have a special direct effect on the infections. So not only all presentation in the patients that we see in the clinic, we know that all human, all healthy individual could be predisposed for the antibody, for the infections. So we should consider to reduce the effect of the SNP that have a direct effect on infections, and we just consider those who have a direct effect from SNP to immunoglobulin level and later on to the effect on the infectious presentation. We know also some SNP can involve in the confounders between immunoglobulin level and the infections, like a herpes simplex disorder that we know EBV or CMV, they have also a specific effect on the B cells, and they can also severe or synergic, have a synergic effect to make a complication of the patients more severe. So we should also find the confounders and find some SNP that are associated with the confounder as well. To just find the main effect of those SNP can predict immunoglobulin levels and those that immunoglobulin levels that will lead to the infectious problem. Don't want to go to the detail, just this is a specific method. So you need to exclude the monogenic, non-monogenic. Then you include also the databases that have healthy individuals, and then you include the databases of the GWAS study that they performed before and antibody levels, different type of infections, and then we make a model, which I think is very specific. I will finish fast, I know, then just a few slides left, maybe 2, 3. So with those SNP, we just find the 57 best SNP significant, which can very nicely find the different population of monogenic from non-monogenic. And nicely, we saw a completely different pattern when you have those HLA regions that are enriched in the non-monogenic patients. They have a low level of the polygenic score, which means this SNP are less. All of those who have a non-monogenic, have not associated HLA regions, they have those SNP that can predict non-monogenic level. So, which means in the cases that we don't find a monogenic defect or a genetic responsibility for those cases, we have 2 different patterns. Some of them can be predicted by HLA region, some of them can be predicted by these SNPs. What we should do in future, we know that the HLA typing is not only specifically should be done for the patient who are non-monogenic, maybe HLA typing has some roles, those ones that we saw that are higher in the monogenic patients, maybe they can also predict a little bit better the phenotype of patients who have, for example, severe phenotype in a specific family. We think that HLA typing was also important to the cases from the familial cases that we have a genetic finding, but they have a clinical discrepancies. And we should also perform some epigenetic studies and wiring tests to further define what's going on with the cases that are not predictable, but HLA and the polygenic score studies. At the last, I want to thank all of my colleagues, mainly my colleagues from the Sweden, Professor Hammarström Group and Professor Pam Hammarström Groups, and also my friends from Tehran University Medical Science Research Center for Antibody Deficiency, Professor Abolhassani and Professor Nima Rezai, and all the specialists that we have in our network in Iran that are supporting us with the follow-up of the patients and treatment. All of my friends in our CVID, thanks for your attention. I'm happy to take your questions.
So, one brief question, if there is, because we are running out of time. So, is there any? So, really not some questions, but thank you very much for the presentation.
I want to also invite Professor Peter Sendler, who is will present for us some studies from the ADA1 and IDA2. We'll be all happy to hear your studies.
Good afternoon. I'm coming from a very small country, and